Hello, and welcome to day 2 of the National Press Foundation Covering Rare Diseases Journalism Fellowship Program. For our first session, news from the world of genetic therapy research offers lots of exciting, hopeful signals about how technology and innovation can improve human health. Stefano Rivella is at the forefront of research centered on blood disorders like sickle cell anemia, which our speaker yesterday our third speaker yesterday opened up that world for us. Stefano Rivella is a professor of pediatrics at the Children's Hospital of Philadelphia. He is also the scientific director of the Comprehensive Center for the Cure of Sickle Cell Disease and other red blood cell disorders. You can read his long and extremely impressive bio on our website at nationalpress.org. But I learned about Stefano Rivella's work through a fascinating article that I came across about novel in vivo gene delivery tools for various diseases. I think one way of also describing them would be a one-shot cure for many of the diseases that we'll be exploring this week. Not only will he explain to you what that means, but Stefano Rivella will also brilliantly enlighten our fellows about the quest for equity in rare disease treatment. Stefano, thank you so much for joining us today.
Thank you, Rachel. You're extremely kind, and introduction really makes me really humble because I'm a scientist and I'm not used to to hear these kind words about my work, but it's very, very flattering.
So I've interviewed and worked with scientists literally around the world, and I really appreciate your approach to this. So before you get into your formal presentation, could you start by telling us about your recent trip to Malaysia and the conference that you went to there? And I think that'll help us set the tone for your conversation.
Absolutely. I actually eventually have a slide during my presentation talking about this trip I had in Malaysia. But the bottom line is that when I started working on gene therapy, I was obviously very much interested in trying to find a cure for diseases such as sickle cell anemia and beta thalassemia that I will provide a little bit more information when I start my presentation. But the bottom line is that the work on gene therapy or the quest to find a cure for sickle cell anemia and beta thalassemia started, I would say, more than 20 years ago. And at that time, as you probably know, the only way to treat these patients was through palliative ways of keeping them alive, like blood transfusion. Maybe there was some opportunity to do some bone marrow transplant, but there were no therapies. Like a definitive cure or a gene therapy was just a concept that people were discussing about. So in 2000, I somehow among other people discover a new tool, a gene therapy tool that immediately sparked a lot of interest because, at least in mice, it showed that it was possible to cure diseases such as sickle cell disease and beta thalassemia. And I remember that around that time, we were talking about 2000, the year was 2000, that a lot of people, including patients, were talking and asking me, and I was going through seminars and invited to presentation, etc. And there was a lot of interest through the community about these novel therapies. Well, this year I went to the Thalassemia International Federation. My trip was in Malaysia, and this is a federation that really focus on the federation focuses on basically trying to provide support to the patient and how to be treated, where to find treatments, how to harmonize treatment between different nations, how to provide innovation in a way that patient can be treated better in countries where maybe information about the disease and new treatments are not yet available. And I was really impressed, in a way, in a negative fashion, by the fact that all the questions were not really about the novel therapies, why this gene therapy works better than other gene therapy, but we're about basically how can we be treated? What is going to be available to us to be treated? Because there was clearly an understanding that now the novel therapies are available. The price was too high for the majority of the patient living in countries such as Malaysia. And the patient were really focusing on the fact that we should have cheaper treatments, we should have better treatments, we should have at least those basic drugs that are now available in other countries, but at a much lower price. And so to me, that was really, I was aware of the situation, but somehow being in a place where more than 1,000 people attended the conference, and most of them were patients, and most of them were basically repeating and asking the same question. And there is a struggle. There is clearly a struggle about the difficulty of providing these novel therapies to this patient. And I think this is really the challenge of somehow, is the challenge for us as a scientist, but especially for industry, how to translate in a way the possibilities to a real application for these patients. So I should start my presentation at this point, I believe. And let me share my screen. So this is the title as in the program. And I want to start with my disclosures. I just want to say that I'm all the science that we'll present today has been supported by federal or local grants. I do work with industry normally as a consultant, try to help them to design clinical trials. And this looks like a long list, but I really like to show all my consultation in the last 5 years, even if they are not anymore active, for people just to be aware. But I just want to clarify that all the information that I will share with you today were not supported by industry. So let me start with a little bit my background. So my name is Stefano Rivella. I'm a professor of pediatrics at the Children's Hospital Philadelphia. And also I have the honor to hold the Kwame Hoini Frimpong Chair on Sickle Cell Anemia. Dr. Frimpong was an eminent doctor and working on sickle cell anemia for many years at CHOP, who established eventually an hospital in Ghana after he retired, with the idea to expand the treatment for sickle cell anemia also in his original country, where the infrastructure were not adequate. And unfortunately, he passed away a couple of years ago. He was a dear friend, but I still been very much influenced by these ideas that what we do should be really, the science we do eventually should be applicable to all the patients on every single country where they are. So I'm also the leader of the RNA Gene Therapeutics Group at the Penn Institute for RNA Innovation, that's been recently established by Drew Weissman, who recently won the Nobel Prize for his work on the RNA delivery system that eventually led to the development of the COVID vaccine. And I'm the scientific director of this center called QRED that I will discuss a little bit in a few slides, which is a novel center for the treatment of many diseases, including sickle cell anemia. So I want to start with a brief word of hemoglobinopathies. So hemoglobinopathies are really an umbrella, name that defines as an umbrella three major diseases. And these are diseases where basically there are mutations in at least in one of the two genes that are responsible for the production of hemoglobin. So hemoglobin is basically a protein that's present in all our red cells. And the function of hemoglobin is really transport oxygen. So it binds oxygen in the lung and deliver oxygen into every single cell of our body. So there are two flavors of hemoglobinopathies, those that have mutation. So the hemoglobin is made of two proteins called alpha and beta. And there are hemoglobinopathies can come in two flavors: those that have a mutation in the beta protein, beta globin protein, and when there is a specific mutation in a specific amino acid of this hemoglobin, we can have sickle cell anemia, which is characterized by a different shape of this hemoglobin that basically creates rods, very, very stiff rods inside the red cells. And that's why the red cells eventually look like with this sickle shape. And the main consequence is that these red cells, being very rigid and very not flexible, they can basically create vaso-occlusive episodes that can be extremely painful and lead to stroke-like symptoms. So you can have basically stroke-like symptoms in muscles, in your brain, and in many other organs, and can lead to damage. And to prevent that, patients normally require medication, require blood transfusion, require also pain medications. And there is a lot of stigma about this disease, because a lot of patients, unfortunately, require strong pain medication, and many doctors are not aware of this disease, may think that the patient are abusing of these substances. Well, really, they have a major pain crisis. So another kind of disease is called beta thalassemia, and in this case, there are like more than 400 mutations in the beta globin gene, but all of them basically lead to a decreased production of the beta component of hemoglobin. In this case, we have a relative excess of the alpha chains. And when you have too many alpha chains and too little beta chains, these alpha chains basically interfere with the quality of the red cells. The red cells die very quickly, and basically you don't make enough red cells. And in severe cases of beta thalassemia, patients also require chronic blood transfusion for survival because they don't make red cells. On the other spectrum, we have alpha thalassemia, where basically we have the opposite situation. So we have mutation in the alpha globin genes. And in this case, we have an excess of beta chains. And we have a similar situation where, again, the imbalance or the excess of beta versus alpha also affects the quality of the red cells. And in this case, in severe cases, alpha thalassemia is also associated with a chronic blood transfusion. I want to stress that the alpha thalassemia is kind of like somehow the neglected sister of these diseases, because it's a disease that's less frequent than beta thalassemia and sickle cell disease, at least in the United States. And so there are not many studies on this disorder. However, if you go in Southeast Asia, if you go to Malaysia, to Indonesia, to Thailand, Southeast China, alpha thalassemia is the most frequent disease, congenital inherited disease in those geographical areas. And of course, hemoglobinopathies are not only important because patients, suffer by this mutation, but also because altogether these are the most hemoglobinopathies is the most frequent disorder on the planet. We roughly 300,000 children are born with hemoglobinopathies every year, and mostly in countries where they don't receive adequate treatments. So this is a slide that I mentioned before, where basically I just want to show you here that on the top you can see two papers that were published in 2000, 2001. In the 2000 paper, I was fortunate to be part of the group that discovered the first lentiviral vector, and then I'll discuss this a little bit more later. But basically, it's a system to deliver a gene and try to correct the disease and in this case, in showing that with this system, we could at the time we were able to correct beta thalassemia in mice affected by this disease. In 2001, another group showed that a similar approach was able to basically cure mice affected by sickle cell anemia, but the strategy and the technology was very, very similar. So now jumping from 2001 to 2022, basically we have now the first gene therapy treatment called Zynteglo, which is basically stem out from these two papers. And basically, this is a gene therapy lentiviral vector adding a corrected form of the gene, the beta globin gene, to the patient's bone marrow cells. And you can see that, after 20 years, this is now a reality. But if you look in what it says, basically this at the top of this image, it says basically the price for this treatment is $2.8 million in the United States per patient, which obviously you can imagine very, very quickly how this is going to be a major challenge, starting from many patients in the United States that may not have an insurance that can cover that kind of approach or treatment. But you can imagine how this is going to be impossible in countries like Malaysia. And below, I just want to show you the meeting from which I came back very recently. I was talking before, and where I was presenting some of the innovation that we we discovered in my lab, but where the majority of the question were, how can we afford these new treatments, when they're going to be available, what can we do to make them available to all the patients? So I want to start my journey discussing a little bit how we got to this therapy, and I think it's important to put everything in perspective. So I studied in Pavia, which is a city south of Milan, where I did my training. And then in 1997, I moved to New York City. So when I was in Pavia, I was working on cloning genes, was part of somehow the consortium that was trying to sequence the full human genome to discover new genes and eventually mutation associated with diseases. So in 1997, I wanted to learn more about technology related to a way to cure people. And in that year, people start talking about gene therapy. I have to say that the majority of the people I was talking with, they were telling me that this was going to be a bad investment because gene therapy was not going to be amenable, working, and possible, etc. But in my mind, the structure of human genome was well recognized, was now being defined, etc. And my point was, how can we use this information to help people to live better, especially for those with chronic conditions such as hemoglobinopathies? And so I started working on hemoglobinopathies, and I just want to share with you some information about the structure and expression of these genes, because somehow this basic information will help us also to understand better how we work and why we do the things we do in gene therapy. And I didn't say this at the beginning, but I'm really, really happy if you have any question anytime or during my presentation. If I don't say something that is clear, I say something in a way that's not clear, or you have a pressing question about anything that I'm talking about, I'm really happy if you interrupt me and ask the question. So don't be afraid to do that, please. So going back to the science behind the expression of the what we call the globin genes. So we need to know that, as I mentioned before, the adult hemoglobin, so the hemoglobin that normally is present in people after birth, is called adult hemoglobin, is made of beta and alpha proteins. And here you see the beta globin, the beta globin gene. And here below you see there are two copies of the alpha globin gene called alpha 2 and alpha 1. But during development, it means, from the day of conception to birth, in reality, we don't express, for instance, we don't express the beta globin gene at a very high level. You can see here that, birth is indicated here with this number 1. And basically before birth, we really express the alpha globin gene, but instead of expressing high level of the beta, we express another gene called gamma. And this gene is here, and there are two genes called gamma A and gamma G. And when these two proteins combine with the alpha globin gene, they make something called fetal hemoglobin. And just for you to know, this fetal hemoglobin, the function of this fetal hemoglobin is basically for the fetus is to steal oxygen from the adult hemoglobin of the mother. So in other words, the mother carries oxygen to the body, but also carries oxygen to the fetus, to the baby that's going to be, is going to be born. And the question is, how the hemoglobin from the mother can transfer oxygen to the hemoglobin of the children if they basically they're equivalent. So in nature, the fetal hemoglobin has been somehow invented so that the affinity, the ability to steal oxygen from the mother hemoglobin is higher. So in this way, the fetus doesn't, always has an adequate amount of oxygen for growth. And so before birth, basically, the babies are expressing this fetal hemoglobin, but after birth, the gamma globin gene is basically switched off, so goes down in level of expression, the beta globin gene goes up, and now if you have a mutation in the beta globin gene is when you see the problem. So you may have sickle cell anemia, you may have beta thalassemia. So now, and I will discuss, and probably the CRISPR technology that's been going to discuss also later today, but I will show you some slide. That one way to basically help the children to maintain, to prevent or to cure conditions such as sickle and beta thalassemia is to reactivate the gamma globin gene, so that even as adults, people can express fetal hemoglobin. Even this fetal hemoglobin has a higher oxygen binding affinity than adult hemoglobin. However, has been shown to be productive and helpful for the patients that eventually have no expression of the beta globin gene or mutation in the beta globin gene. So this leads me to, so in other words, when we think about gene therapy, when we think about a way to help patients with sickle cell anemia or beta thalassemia, we can do basically three things. We can try to add back a healthy copy of the beta globin gene to the cells of the patient that have mutation in the beta globin gene. We can basically reactivate the gamma globin gene. So then basically patient, instead of expressing the adult hemoglobin, which is defective, will express fetal hemoglobin, which can compensate for the lack of beta globin expression, or we can correct mutation in the beta globin gene. Obviously, if we correct the mutation, the beta globin gene will not sickle anymore, etc., and so that would be restored to normal function. So the first approach to do gene therapy when I started working in the field was called gene addition. Now we talk about gene editing, etc. We'll discuss in a few slides, but at that time, the technology to do gene editing was not, very efficient, and people didn't know how to do that very well. But they realized that there was another way to do gene therapy that was called gene addition. So the word gene addition really kind of self-explain what's the concept. So the idea is basically to add viruses, and in this case we use a virus of HIV. But the way was modified HIV is basically HIV is made of an envelope and an information. And what is really detrimental to people is the information. And so what we took advantage of HIV, which is a good virus to infect cells, is we replace completely the information of the HIV genome and replace that with the information to make a beta globin gene. And so in other words, we take the envelope, we discarded the letter inside, and we replaced the letter with a new kind of information, so that was able to deliver a functional copy of the beta globin gene into the cells of the patients. So now you need to know that this system is very good. It works. Now several patients have been treated with this technology, and they're cured from sickle cell or beta thalassemia. But this technology also, we need to be aware, has its own limitation. So I try to show here basically that after mixing the cells of the patient with these viruses, a process called transduction. That means basically to deliver this new information through using a virus to the cells of the patient. basically we can integrate the transgene or the curative gene inside the chromosomes of the patients. And this insertion is not precise, it's a random integration. So in other words, we are sending someone to introduce, let's think about like the genome as a library, right? And each library is a different chromosome, and in each library there are multiple books that represent genes. Now, what we're trying to do, we're trying to insert a new book in one of these libraries and make sure that this book is active and working. But also, we don't want to destroy we don't want to integrate the new book in a way that destroys the function of another book, right? And so this is one potential issue when we think about gene addition, because the insertion of this new gene may interfere with the function of another gene. This didn't happen so far using the novel technology for gene addition, but it's something that happened in the past when the design of this vector were not ideal yet. People didn't know how to use them very well, and so that led to some potential issues. So when you think about introducing a new gene, especially the beta globin gene, you need to think about two things. So I like to say, and I mentioned a little bit before this when I was talking with Rachel about the concept of software and hardware. So when you think about a gene any gene present in our cells, in our chromosomes, you need to think that I like to think that the gene per se is kind of the hardware. It's basically the kind of information that is absolutely indispensable to make a protein. So every gene in our cells provides information to make a protein. But in order to make this protein, they also need a software, which is the component of DNA that basically decides where this gene needs to be expressed, when the gene needs to be expressed, and how much of this gene needs to be expressed. For instance, the beta globin gene, as I mentioned before, it's an important gene that needs to make hemoglobin, which is the oxygen-binding molecules inside the red cells. But you don't want to express the beta globin gene in other parts of the body. You don't want to express the beta globin gene, let's say, in your eyes or your skin, right? So the software understands that and basically dictates that the beta globin gene needs to be expressed only in red cells. Another thing we need to know is that the beta globin gene is, together with the alpha globin gene, is one of the genes expressed at the highest level in the red cells. And so not only you want to express the beta globin gene only in red cells, but need to be expressed at very, very high level. So when you think about doing gene therapy, you need to consider these two components. So you definitely want to transfer the hardware, the gene that you want to express in the cells of the patient, but you also need to transfer enough of the software so that the gene will be expressed in the proper way. And so basically, after several years of studies, when I was in New York, we designed a lentiviral vector. This is just a diagram that shows how we clone in this gene the beta globin gene, which again is the software, and then we identified regions of what we call the software. The beta globin gene is the hardware, and then additional regions of the genome of the DNA of human DNA that basically allows the beta globin gene to express at high level. And this was basically another paper where we show that basically using this vector, we were able to increase the hemoglobin levels in animals affected by thalassemia major. So this TH3 indicates basically animals have a very severe form of thalassemia. TH stands for thalassemia. So the TH3 was a model of thalassemia, and the number 3. And this model had basically mutation in homozygosity, so all the beta globin gene were mutated. And you can see that the level of hemoglobin was very, very low. It was not compatible with survival unless you were transfusing these animals. But when we add the vector to the stem cells or the bone marrow cells of these animals, we're able to increase the hemoglobin so that now we were in the range where it was considered transfusion independent. It was not optimal yet, but it was getting really close, and this really excited people about the possibility to use such an approach to cure beta thalassemia. So after these studies, there are a number of papers that were published. I want to show here additional papers where similar lentiviral vector technology was utilized for the treatment of, at this point, even patients with thalassemia. You can see in 2019, some of these papers were published saying, oh, we can use this vector in this patient, and some of these patients actually are cured. And this led basically to eventually approval by the FDA of this vector called Zynteglo from a company called Bluebird. And as I mentioned before, however, as a consequence of these studies and work, and the price tag for each treatment was $2.8 million. And I just want to mention that the Bluebird also tried to get approval for this vector in Europe as well. So this approval, $2.8 million, is a price tag attached to approval in the United States. But they also tried to get approval in Europe. They got approval in human through the agency called EMA, which is basically a similar agency in Europe to the FDA. And I think that there was approved for a price tag. It was less than $2 million. And Bluebird decided that not to pursue the use of the vector in Europe because they considered the price too low. And so, despite the fact that this vector is approved also in Europe, Bluebird is not selling this vector in European countries because they couldn't agree on the price tag in those countries. And again, we can discuss about this later if you have any question.
Stefano,
Yes.
I'm going to jump in at this point because I think you have allowed us to remember something that our speaker, Victoria Gray, said yesterday. She is the first person to receive gene therapy, CRISPR therapy for sickle cell in the world. And she said that her treatment to date so far would have cost $2 million. And there's just no way she could have afforded it. What I'd like you, after you've given us this really solid research basis and understanding of what therapy involving blood disorders is about, could you tell us what it is about this particular set of diseases that is giving researchers like you perhaps a bit of an advantage, or is allowing you to achieve some of these innovations in this? It almost seems like it's exponential. You're learning something and something's happening, and we're coming up with these new therapies. Is there something about blood diseases, I guess is what I'm asking, that is allowing for these innovations to occur?
Yeah, in a way, yes, because the blood is something that we can manipulate better than other organs in a way. If you think about, generally diseases, there are obviously multi-organ diseases that are the most challenging one, but many diseases really focus on one organ in particular. And if you think about it, the organ as a bone marrow, it's a liquid organ, right? And for many years, we realized that this organ is something relatively easy to take out and put it back. It's not like, the heart, or if you think about heart, it's not like something you can take out and put it back. Now we do heart transplant, but it's still, a very challenging and somehow last chance for the patients, where for the bone marrow is something that is being utilized for many years, so taking bone marrow from one patient or from one donor and give it to another patient, or modify the bone marrow aside, like in a petri dish, and put it back. So in a way that working on the bone marrow has been easier than for other diseases. And since beta thalassemia and sickle cell anemia are such a frequent disease, like very, abundant in the sense of number of patients, people were thought that this was a relatively easier place do therapies compared to other conditions, right? And so the fact that you can get the cells, you can manipulate them, like, genetically modify, etc., outside the body in a way at that time was an idea that pushed many people like me to think that there was a place where it was potentially easier to treat disorders such as sickle cell anemia and thalassemia. I don't know if I addressed your question.
I think so. I mean, again, from a journalist perspective, we have to consider that we've got to go back to our editors and say, here's why we should do this story, or here's what's so interesting. And I think for the journalists on this call, many of whom are from the African continent specifically, understanding the potential opportunity for research on blood disorders is important. But I'm going to let you finish because we want to leave some time at the end.
Sure, of course, yeah.
Questions.
So I want to jump to the fact that basically after this, I'm sorry for. So after this news came out about the Zynteglo, around that time, I knew that was, I already knew that the price tag was very high, etc. As a scientist, I really start thinking, well, I feel kind of responsible for this, right? In the sense that I develop a novel therapy. Now, obviously, I wasn't the only one. There were other people involved with the development of this kind of therapies. But I felt like, my drive to do a therapy for these patients, because there were no cures, there were no alternative to their life with blood transfusion and additional medication, etc. And to me, as a scientist, I was really, because of what we were saying, because the blood was amenable, was easy to manipulate, etc. I thought, well, maybe we can do something like gene addition. We can try to put a gene back, etc. But then, when the price tag of 2.8 million come out, basically, I would say personally, I felt very frustrated because I felt like my science, in a way, you need to know that the science is done in a way that centers that we invent new things. The institution may have an intellectual property on the invention you generate. They normally sell this license to a company because it's very challenging to develop these technologies and bring them to the patient through the institution. Can be very expensive, etc. So then I start thinking, is there a way we can control more of these? Can we find a way to sponsor this kind of application through the institution where we're working on to bring, I wouldn't say a cheap product to the patient, because it's not yet possible in the sense that, we won't be able as an institution to cure all the patients on the planet, but at least to provide some kind of structure that can help patient to see how we do things, and hopefully also help companies to provide a better quality and cheaper product to the patient. So around time I joined CHOP in 2015, I started working with a group of clinicians to establish a new center called the QRED Center, here described here. And the idea basically was, can I work with clinicians? Can I work with the core facilities at CHOP, where I work now, like making the vector ourselves? Is the vector we can make in our facility can be cheaper than the vector that's made outside and made by a company? Can we do things here, at least for a small group of patients, where we can deliver these these therapies in a cheaper way? And so we established this center, and basically we were able to bring a novel vector that I made when I joined CHOP in 2015, that has a higher potential for treatment in the patient, etc. And now we have an FDA approved for this new vector in my center, and we are going to start a clinical trial soon. And so far, all the trial has been sponsored by CHOP. I'm not saying it's going to be cheap. I'm not going to say that this is the way to treat all the patients, unfortunately, because We can talk in detail how much it costs to make a vector, how much it costs to treat a patient with the American insurance, etc. But at least we will be able to treat a few patients in our center with a novel vector we made with a definitely lower price than the company can afford, the company can present now to the patients. Now, what's going to happen after this, I can't control it because, as I said before, the IP doesn't belong to me, it belongs to CHOP. And CHOP obviously is interested also in recovering some of these expenses that has been used to make this trial. But I really hope that whatever company will come through will have in mind that our goal is really to provide a novel treatment with a much lower price. But let me move now to also very briefly to the CRISPR technology, because this is another thing that people are utilizing now for the treatment of beta thalassemia, sickle cell anemia. And I just want to briefly remind you that the CRISPR-Cas technology is a technology that was identified several years ago, studying bacteria that make yogurt. In other words, at that time, people discovered that even bacteria that were utilized to make yogurt can get infected by viruses. These are called bacteriophages, which are basically viruses that infect only bacteria. And then in trying to characterize in that relationship, they realized that the bacteria makes this protein, one of them is called Cas9, that basically is able to recognize the DNA of the virus that infects the bacteria and destroy this DNA, preventing the infection. And so Charpentier-Doudna, that won the Nobel Prize eventually to understanding how to use this system to cure mutations or to modify the DNA of patients, realized that this system called Cas9, CRISPR-Cas9, can be utilized also in human cells to do something that could be therapeutic. And so the original system is called the Cas9, CRISPR-Cas9 system that basically target DNA, but basically destroy sequences. But now we have more advanced systems like base editors or prime editors. Without getting too many details, these systems are more advanced in the way they can actually modify the DNA in a way that basically we can revert mutation and so correct mutation in the human genome. And so with this approach, now there is a new product that's called Exacell by a company called Vertex. And in this case, what they do, they basically target a sequence called BCL11A in the genome. And when this sequence is basically destroyed, there is increased expression of the fetal hemoglobin. If you remember in one of my original slides, I show you that basically the fetal hemoglobin is the one that goes down, the beta globin is the one that goes up. But if you basically block this gene BCL11A, you can completely revert this process. So the beta goes down, the gamma goes up, you make fetal hemoglobin. And now this patient basically, it's been shown that more than 40, 50 patients now can express high level of basically hemoglobin or F cells, also indicated here, after treatment. And in this case, they are rescued from beta thalassemia, sickle cell anemia. So I just want to mention that this is also another approach to cure patients with sickle and thal. It's expected that this product may be approved at the end of the year in the United States. So I don't know what's going to be the price tag for this product. I believe it's going to be cheaper, and I hope it's going to be cheaper than the Bluebird product, but I don't think it's going to be very cheap. So it will be interesting to see how approval of this product will affect the market and the way the people have access to this treatment.
Stefano, I'm going to jump in here only because, unfortunately, we're coming to the end of our hour, and we've got a couple of really important questions from a couple of our fellows. We're going to be able to post your presentation online, so we'll have access to it. But I want to refer you to Pascalina Kabi. She's one of our fellows from Lesotho, Uncensored News there. She has a very important question to pose to you. So Pascalina, can you jump in?
So I'm wondering if it's possible, like in countries like Lesotho, the issues of HIV medication, they are offered on a subsidy. So I'm wondering if African countries can apply the same subsidy measure to advance medication to rare disease patients, and if you think, Stefano, that this in any way can help ease the burden, the financial burden on patients and caregivers. Thank you.
Yeah, I think it is a great question. If I understand exactly what you're saying, I think the key here is to understand how can we bring those medication in countries such as Africa, Southeast Asia, and access them and how to pay them. Is that really your question? I just want to be sure I understand correctly.
Yes, it is. But added to that is in terms of payments, for some people, it is very difficult to raise money to pay for medication. So I'm wondering if there can be some sort of an arrangement where government says maybe we offer a 10% or 10% subsidy on this particular medications for patients.
Yeah. Okay, I get it. So I personally, and again, I don't hold any relationship with companies in the sense, work with them as a consultant, but not equities or anything else. But my personal opinion would be, there are countries that can afford some of these drugs, and they can pay a little bit more, right? And there are countries where this is not possible. And so my personal opinion would be, or my personal suggestion would be, why don't we let this country or teach this country how to make these medications? So then, first of all, the cost would be much lower doing creating this drug in countries where the cost would be lower for obvious reasons, and support this with some of the the net income that comes from richer countries. So in other words, we can subsidize. My way to subsidize this would be teaching and allowing countries to make their own medication, right? To establish facilities in those countries that would hopefully make these drugs a lower price, and subsidize these, so that then if we don't subsidize directly the drug to each patient, we may be able to create drugs that are much cheaper to start with in those countries. Again, I'm not an economist, so what I'm saying should be taken with a grain of salt, because I don't know the details of all these things. But to me, that would be a logical thing to do to help people in this country to get those medications.
I think there is some precedent for this, Pascalina, that we could explore and you could explore, in that I believe I just read something that said the malaria vaccine is actually going to be produced in Kenya. Don't hold me to that, but I truly believe I saw that recently. So I think there is movement towards having this happen. I think as a nod to our speaker, we must hear from Manuela Carrari of Cosmos magazine in Italy. So Manuela.
Hi, thank you so much for your talk. I have two questions. One is whether it's possible to detect those diseases in fetuses, since the beta gene is not expressed, or it's not expressed as much as the other two. And the second question is, what exactly makes these therapies so expensive, so much more expensive than other conventional drugs?
Right. So
thank you.
Yeah, absolutely. Thank you for the question. So first of all, yes, it's possible. I for question of time, I didn't go in the detail of prevention medicine, which is very, very important. For instance, in Cyprus, they basically abated the number of people born with beta thalassemia by prenatal diagnosis. So that's one approach that I think probably would be the ideal one. Of course, there are ethical implications in the sense that, you need to be in a place where abortion is considered legitimate and acceptable treatment for women, and that's obviously can be a limitation in some places. And of course, the other issue is being able to bring those assessment, these technologies also in those countries where not available yet, because we can think about assessing for beta thalassemia, sickle cell anemia, which is obviously very important because it's a frequent disease. But if you start looking at many rare disorders, the panel is much larger than that, right? And so the question is which disease to pick and invest into it. So I think there is a movement in going that direction, and I believe that would be a good thing to do. And the second question you have about the cost, so that's technology based in the sense that, for instance, the way we do now our lentiviral vector, the one we're trying to use a CHOP, is made in ways that is cheaper because we identify ways to do the production, which is the very expensive part, in a way that we can treat more patients per amount of vector we generate, right? And so if we get to the point that we can abate that cost 10 times or 20 times, you can imagine already that $2.8 million is going to be $280,000 million, right? $280,000. And so, again, it's still very high for many countries, but we're going in that direction that basically we can abate the cost already 10 times, right? And if we can get that to the point that we abate it 100 times, then we're going to talk about costs that are going to be much more affordable. And again, with the system that where maybe we can generate these drugs directly in the country where the cost of making this drug is lower, maybe affordable one day. Again, I'm not talking about something around the corner, but that's what I wish for, basically.
We could probably take two more questions, and I think we'll run a little bit longer than our hour. Stefano, I hope you can stick around with us.
Oh, yeah. And I actually only have 3-4 slides, 4-5 slides to finish, but so it may not very long, but I'm happy to take the question now.
Well, we'll take one more, then we'll let you do your slides, and then we'll wrap it up. And this is from Julia Matro of Mother Jones, and she asks, wondering if the rise of rampant disinformation on mRNA in the past few years has changed how you have spoken about it as a scientist.
Sorry, can you repeat the last part of the question?
She's wondering if the rise of disinformation on mRNA in the past few years has changed how you have spoken about it as a scientist.
Yes, definitely. And maybe I can answer this question with the next 3 slides.
Okay, great.
Is that okay? Okay, so I just want to go to basically directly going into the story of the RNA. And what I want to do is just to mention that when we talk about a gene therapy right now, the major limitation, as I mentioned before, is the building of the vector, making the vector, doing the transduction in the cells of the patient. But the other thing is also that you need to do this it's called ex vivo. So you need to take the patient cells outside the body, do the manipulation, could be gene editing or gene addition ex vivo, so outside in a petri dish, and then put them back through myeloablation. And this is really a challenge because increase not only the manipulation, the viability, decrease the viability of the cells that you're using for the manipulation or for the infusion, etc. But the myeloablation also comes with loss of fertility. So every time you need to do a bone marrow transplant, whatever reason you need to do, the patient is affected by the quality of life of the patient is affected tremendously because these reagents that we're using now basically are killing the germinal cells in both women and men. And of course, the question is, can we use a different technology to do these gene therapy approaches? And so here, just make a schema how we can use basically the COVID technology, which is made of lipid nanoparticles. So basically, if you think about the COVID vaccine, these are basically fat molecules, right? That basically are fat balls that contain a piece of RNA inside, and then they can be delivered, in this case of the COVID vaccine, they're basically an injection in your muscles. and then to generate immune response against the COVID virus. But the point is, can we modify these to manipulate this LNP, these fat balls, and sell them and send them directly to the bone marrow? And if we can do that, you can think about novel therapies. You can think about RNAs that go to the bone marrow to improve the quality of the bone marrow cells. You can send this fat ball to the bone marrow to kill the stem cells. And this could be an alternative way to do a bone marrow transplant, less toxic than what we do now. But obviously, can we also send these RNAs to correct the hematopoietic stem cells? So that's basically the idea. And I just want to mention that basically in the last few years, we've been shown in this publication that I mentioned before, with the contribution of several members of my lab and the collaboration with Drew Wiseman, we basically identify a way, and we show as a principle, that we can deliver these RNAs into the bone marrow of mice, and we can modify them so that if initially these bone marrow cells are normal, we can modify them that became fluorescent. And this fluorescence, so you can look at the microscope of these bone marrow cells, and now they're basically red, in the sense express a color red. And this can be done only through genetic manipulation. But this genetic manipulation is mediated by this RNA that is basically included in these lipid nanoparticles. And just to show you basically the results that we achieved through this approach, we can see here that the majority of this is a very technical if you want, but the bottom line is you can see that now between 60 and 70% of all the cells that come out of the bone marrow of these animals are genetically modified by this approach. And my last slides is pretty much showing you that, my last two slides is to show you that if we can do these and try to target with an mRNA the mutation in patient cells from a sickle cell patient, we can correct the mutation. And so we can, in this case, it's not being done in the patient body, but in these cells treated outside the patient body. But basically, if we do this, we deliver this RNA to cells with the mutation and with the sickle cell mutation. Now, the hemoglobin that we make out of these patients is is normal. Is, you can see 80% of this hemoglobin now is very high. And if we try to look for sickle cells in the cells that come out from these genetically modified stem cells, you don't really find them. So you can see here in the patient cells that have not been modified, you can see by these triangular arrows, you can see a lot of sickle cells. But now if you treat these cells with this technology we invented, now you don't see basically sickle cells in the coming out from these modified stem cells. And so basically this technology is very powerful. And my conclusion, in a way, are these. So I think we can say that gene therapies are now a reality that potentially can benefit a subset of patients. And this has been done by lentiviral vectors and soon can be done also by gene editing. But of course, the limitations are very high cost, and current myeloablative regimens are really, as I mentioned before, come with taxing consequences for the patients. So I think the in vivo gene therapy that we developed, and I want to mention that it requires a lot of work before we will be able to do this in patient. We overcome some of these limitations associated with the current myeloablative regimens. So these has the potential to cure and to many patients and to further advance the field of gene therapy. First of all, because we can eliminate or simplify the myeloablative regimens, or if we can directly do in vivo gene editing, there is no requirement for myeloablation. So the patient will not need to go to the hospital. They can get an injection by a nurse, and this can be done in any country without sophisticated, specialized clinical center to do the gene therapy. And if you think that right now the injection for the COVID vaccine is $5 per shot, even if the cost of these therapies, again, unfortunately, I won't be the one deciding the cost, because as usual, there's going to be companies working on this technology to bring them to the patients. But even if you think even the cost would be 100 times higher, we are talking about $500 versus $2.8 million now with the current therapies. So I think this is the future. I hope that the company in the future will understand that this is going to be a technology that can help the patient, but especially a technology can be amenable to patients in many countries where the sickle cell and beta thalassemia are unfortunately present. So I just want to finish my presentation with this additional information so that we can discuss more any question you may have. And I don't know if I basically addressed the last question that Rachel raised to me with these last slides.
Yeah, I think so. We are fortunate to have now this relationship with you so that the journalists can reach out to you for more information. But I do want to allow one more fellow to join us because she does have such good questions, and that would be Rupsa Chakraborty of the Indian Express. Rupsa, you can finish us up by asking Stefano your questions.
Yeah. I have two questions. My first question is, does this gene editing exhibit comparable efficiency in adult patients when compared to its efficiency in children? My second question is, talking about the clinical trials, will adult patients be part of this, or it will be targeted only for children?
Right, that's a good question. So again, I don't want to raise too many expectations in the sense that this is not a technology we can still use in a clinical trial. So there are a number of things that need to be done because as any novel technology need to be tested in many ways before we can really show that it's not only working, but also safe, obviously, because, you can imagine we are now directing something to do gene editing directly in vivo. So we need to know if other organs may be affected, the germinal cells will be affected, if there are going to off-site effects. But assuming that all these problems are not present in the sense that the approach is safe, I think it provides a much better tool to treat patients, not only children. Because right now, the current limitation for gene therapy is the myeloablation, right? And as you probably know, as you get older, you get, unfortunately, more, you acquire more limitation due to the fact that, for instance, the iron overload in thalassemia or due to the chronic transfusion may affect your heart and liver and other organs. And when we think about myeloablation, that's a problem. If you have already damages in your organs as you get older, you're not amenable to myeloablation. But if this system is going to be a reality, that's not a problem because there's not going to be myeloablation. So you theoretically can cure patients no matter what is their age.
I will end by recalling something that you just said and that you said during our original conversation, and that is moving from a scenario where treatments cost $2 million to the ability to have this technology and this treatment delivered in a shot in a clinic by a trained clinician and it will cost $500. I believe that the conversation around health equity on a global space need to be led by researchers like Stefano Rivella of Children's Hospital of Philadelphia. And thank you so much for joining us today. And I hope that you will be open to hearing from the journalists in the Covering Rare Diseases program as they begin to develop their projects. So thank you so much for being here today.
Absolutely. Thank you so much.
And now we have 25 minutes, you guys, before we come back for the next session. So thank you. Stefano, thank you so much. We're going to post your presentation. As I say, I think people like Rupsa are going to be calling you like tomorrow and saying,
yeah.
I want to do a story.
Absolutely.
And you can help us out. So thank
Thanks
you.
a lot. Bye.
Yeah, I'll be in touch.